Okay, so protein synthesis is what we're going to be talking about today. There's a few steps. Step number 1, copy DNA code into a messenger RNA molecule. And here's a picture that we could use to represent that. So this is a chromosome, and the chromosome is made up of 2 backbones and those 2 backbones, does anybody remember what the 2 molecules are in the backbones? Yeah, Jonathan?
Deoxyribose sugars and phosphates.
Good. So 5C and P. Phosphate is represented by the P, and 5C is the carbon. The third piece of the nucleotide, remember the nucleotide is made up of 3 parts, is what? What's the third part of the nucleotide? The third part of this piece, so there's 2 parts to the backbone, 5 carbon sugar and phosphate. The third part is what? The rungs of the ladder. What's that called? Nitrogen base. Good. Those nitrogen bases are made up of A's, C's, T's, G's, and in a specific sequence. So this is all going on in the nucleus. But when the DNA unravels, there's weak hydrogen bonds holding these together. So there's a hydrogen bond. That hydrogen bond is just about to break. Here it is breaking. So the hydrogen bond holds the nitrogen bases together. When those hydrogen bonds break, the DNA unravels and the backbones are exposed, revealing the code. When that happens, a chunk of messenger RNA is built, is created using that DNA code. So this is the mRNA molecule being formed right here. That mRNA molecule is specific to the sequence in the DNA. And here's how it works. The mRNA, just like the DNA, has a 5 carbon sugar and a phosphate for the backbone. The nitrogen bases are a little different. C will bond to G in mRNA, just like in DNA. G will bond to C, so these 2 are okay. T bonds to A, just like in DNA. C with G, and that's okay. But right here, where the A would normally bond to a T for thymine, RNA has no thymine. Instead of the thymine, it has a uracil molecule. So U is uracil. And that's right here. So that uracil replaces the T. So wherever you see an A on the DNA, it would normally bond to another A over on this backbone, but since this is an mRNA strand being formed, it's a uracil. Okay? So A bonds to U. Okay. Well, that is a chunk of messenger RNA, and here is the nuclear membrane. That mRNA is exported out of a hole in the nuclear membrane. So this is the wall of the nucleus here. And here is, it's called a nuclear pore. And that mRNA is threaded out of the nucleus on its way into the cytoplasm. Okay, that's step one. I'm going to turn the page for step two. Is that okay? Raise your hand if you're not done copying this down. Okay. We'll post this online so you can finish, okay? All right. So the mRNA, so step two. mRNA matches up to individual tRNA molecules to create, or actually to attach amino acids to a growing protein chain. Okay, well, let's see how that works. So here, leave a little space, leave a big chunk of space above the mRNA. So this is the mRNA. So the code on the mRNA, here's the amino acids on the mRNA. mRNA, by the way, is not double-stranded, it's single-stranded. So the nitrogen bases are exposed. And let's say that this is A, U, G, C, C. Let's say that that's the code on this chunk of mRNA. Every 3 nitrogen bases is called a codon. Codon. Write that down. Does your book talk about codons? No? Okay. Well, you'll learn about them when you get to high school. You'll want to remember that. So, if this is, I'm going to just move this label here. So this is mRNA. C A U G. So there's one codon. There's another codon. And let's make one more. C, U, A. So here's what happens. The messenger RNA docks with a ribosome. And the ribosome kind of looks like a roasted chicken. In every drawing I've seen in any book, kind of looks like a roasted chicken. Here's a drumstick here. Well, here's what happens. 3. Here's what happens. The messenger, so let me label this. There we go. So here's, I'm going to make this exaggerated. Doesn't look like a roasted chicken anymore, but I want to include all this. So this is the ribosome. Ribosome. And here's what happens. The transfer RNA, the tRNA, is named transfer RNA for a very specific reason. And that's because it looks like a T. An upside down T. And that T has on it nitrogen bases that code for a piece of the DNA or the mRNA. So this is the tRNA. tRNA. Well, if this right here, the AUG on the mRNA, if this is AUG, then the transfer RNA that fits to that is a U, because that goes with the A, C. That's right. So that is coming in to dock with this codon right here, first 3. The next tRNA, let's draw it in. This docks on here. The next tRNA has the code of G, G, U. And that's docking with the help of the ribosome. Well, there's one piece that I have not mentioned yet, and that is that attached to each amino acid, or attached to each tRNA, is an amino acid. Here is one of the 23 essential amino acids here. Here's another amino acid attached to this tRNA. We'll make it round. This is an amino acid. And those amino acids are being joined together. And let's draw another tRNA over here that has just come in and delivered its amino acid. Here's another tRNA that has just docked and dropped off its amino acid. So it's coming in here. The tRNA is coming in this direction. And it's leaving in this direction. So it's going away in that direction. And it's coming in and delivering amino acids. And here is the amino acid that used to be attached to this tRNA. I'll draw a little ghost amino acid here, a square one. That one has just dropped off the amino acid that's coded for by this codon right here, the UGC codon. So this is amino acid 3. This is amino acid 2. And this is amino acid 1, let's say. And what happens is that there's a growing chain of amino acids that forms the protein. Label this protein. Okay. So as these transfer RNAs come in and dock with the ribosome, and the mRNA is being threaded through it, they deliver their amino acids based on the code in the messenger RNA to build the protein. Okay, does that make sense? Step 3. The unique protein is made. And that protein is made up of amino acids. Amino acid chain. There's one rule. One rule. One gene equals one protein. And there's about 30,000 different proteins in the human body. They have many functions: repair, growth, defense, reproduction, metabolism, and messaging, just to name a few. Okay. Proteins are super important in every living creature on Earth. And genetics is really about the production of proteins. How are proteins built and what do they do? Okay? All right.